Around 1950, Chargaff noticed that the frequencies of adenine, guanine, cytosine, and thymine varied across species, and that the frequencies of G was the same as C and A was the same as T. A chemist around the same time named Rosalind Franklin, who generated DNA crystals and shot X-rays through them to study their structure. The X-ray pictures showed a repeated pattern, which meant that the DNA was something similar to a helix. Based on these discoveries, Watson and Crick were able to discover the features of the structure of the DNA. They discovered that DNA was not a single helix, but an antiparallel, double-stranded, right-handed helix, connected with nitrogenous base pairs that are found inside. The nitrogenous bases are connected through hydrogen bonds that hold the DNA strands together. Watson and Crick's original model proposed that there were two hydrogen bonds between each base pair, but we know today that G and C form an additional bond. The backbone of the DNA is made up of repeating units of sugar and phosphate groups which make up the outside of the helix. The oxygen on the phosphate group is negatively charged, which makes the DNA negative. Another force that affects the stability of the DNA double helix is the van der Waals forces, which is responsible for the base stacking interactions that bind each base pair to the next sequence. The double-stranded DNA contains a 5' end and a 3' end. The 5' end of one strand aligns with the 3' end of the opposite strand. This determines how each strand is replicated. Each helicase has a handedness, which is a property that describes how their grooves are oriented in space. The twisting of the DNA double helix bases create a wider gap called the major groove and a narrow gap called the minor groove. The grooves are really important binding sites for proteins as they maintain the DNA and regulate gene activity. Proteins are more likely to bind to the major grooves as they have more access to it than the minor grooves. To differentiate DNA from RNA, we can look at the sugar molecule in the backbone. DNA has deoxyribose sugar, while the RNA has ribose sugar. This means that the DNA will only have a hydrogen in the 2' carbon, while the RNA will contain a hydrogen as well as an oxygen in the 2' carbon. The carbons on the sugar are really important because they tell you what attaches to them, which helps when creating the nucleotide chain. Other carbons that are found on the sugar molecule are the 1' carbon is where the base attaches, the 5' carbon is where the phosphate attaches, and the 3' carbon is where the next nucleotide is going to come and make a bond to the next 5' phosphate. DNA can either be right-handed or left-handed. To identify which one it is, look at the backbone of the DNA. If it's going up to the right, then it's right-handed. If it's going up to the left, then it's left-handed. Keep in mind that the normal form of DNA is a right-handed helix. Some examples are shown on the screen.